Well, today I'm going to deal with the topic on cell. Uh, Cell, it's a basic structural and functional as well as a biological unit of a living organism, and it was first observed and coined as a term cell by Robert Hooke, who has observed when he has taken a thin slice of a cork. He has observed it under a microscope where he has found rows of empty boxes which appear to be pores or boxes which are arranged in rows. And since there was no microscope, properly developed microscopes with high magnification view or high resolution, he has under low magnification only, he has observed it and coined the term as cell. Later, there were many theories being proposed based on a cell to know what actually it is. So, based on that, they have come, Rudolf Virchow, along with his colleagues and many other scientists, have come to a conclusion that cells arise from pre-existing cells. That is, a cell which is already existing, that is, which is already present, helps in causing the division of the further cells and gives rise to new cells. So, as I've already told, cell is defined as a basic structural, functional as well as biological unit of every living organism, and it is mainly basically broadly classified into two types, that is the prokaryotes and the eukaryotes. Prokaryotes, they are the structures mostly under prokaryotes. The organisms which come are the bacteria and the archaea group, and whereas in the eukaryotes it is the all the cells present in the plants, animals, and also in various other organisms. So coming to the basic differences between eukaryotes and prokaryotes. Since the term itself suggests eukaryotes, eu means it is properly developed karyon. Karyon is nucleus. And prokaryotes, it is a pre-karyon. That is improperly developed or nucleus which is in a stage of still developing nucleus. And coming to the structural and the functional, based on these structures, presence of the structures as it functions. So the differences are based on the presence of the structures in both the cells. In eukaryotes, it's a properly defined nucleus can be seen with which is being surrounded by a nuclear envelope, and this nucleus contains chromatin, which is a genetic material which is arranged in a form of circular DNA. Whereas in a prokaryote, you don't find any nucleus, but the chromatin material, it is being disorderly arranged and it is usually in a linear form, and there are even certain chromatin material or the genetic material which are being away from the nuclear region, that is from the center region, and these are known as the extra chromosomal DNA, that is known as plasmid. And so, coming to the other cytoplasm which is a fluid-like gel in which the cell organelles, they are being suspended. And in this cytoplasm, we can observe the other organisms, other cell organelles, that is mainly coming to the organelles which are present just adjacent to the nucleus is the endoplasmic reticulum. This endoplasmic reticulum is a membrane-bound organelle and is of two types, rough endoplasmic reticulum and the smooth endoplasmic reticulum. Rough endoplasmic reticulum, it is so called because of the presence of ribosomes which are studded and gives an appearance of a rough appearance. Whereas the smooth endoplasmic reticulum has no such other organelles embedded on it, that is ribosome, so it is named as smooth endoplasmic reticulum. And this endoplasmic reticulum organelle, which is a membrane-bound organelle, is not seen in the prokaryote cell. Coming to the next group of organelles present in the cell in the eukaryotes are the ribosomes. These are the structures which are minute structures, and they usually help in the synthesis of the protein, and they are known as factories of protein since they synthesize these proteins. They are usually found in the prokaryotes since they are the basic structures, proteins which are the building blocks of a body. So we find them even in the prokaryote cell. And coming to the other structures, other than that, the next important group is the Golgi apparatus, which helps synthesize protein to be transported to the other organelles, where we do not find it in the prokaryotes. And there are even membrane-bound limiting organelles as well as those which are not bound by membrane, that is, which are not being covered by a cell membrane or a plasma membrane or a cell envelope. There we find the other structures are lysosome, peroxisome, microtubules as a cytoskeleton, and the other structures which are found here in the prokaryotes is other membrane-bound organelles cannot be found except the ribosomes, and the other structures which can be found extracellular, that is outside to the cell, is the cell envelope, that which constitutes the cell wall, plasma membrane. Together they are called capsule. Whereas in the eukaryotes, you find a cell membrane, plasma membrane, and a properly fixed centrioles and centrosomes. This finishes the differences between the eukaryotes and prokaryotes. Coming to the levels of organization of a cell, usually as we have already discussed, saying that a cell arises from a pre-existing cell. Once a cell is formed, these cells, they group into structures known as tissues. So these cells could be nervous cells, nerve cells, muscle cells, epithelial cells, any intestinal cells. And further, they constitute these group of cells together, they constitute a tissue. This tissue could be a nervous tissue, muscle tissue, or an epithelial tissue. And then this tissue together, it constitutes and gives rise to a new level of organization known as organ system. Suppose it can be a nervous. These organs could be brain, liver, or skin, or any GI related organs or anything. These organs together, they constitute a system, an endocrine system or a cardiovascular system, a nervous system. It could be. So coming to an example, giving an example, it is. Considering a nerve cell, this nerve cell, it groups into these nerve cells, they group into a tissue known as nervous tissue. This nervous tissue groups into an organ known as the brain, and this brain, it constitutes the nervous system. It could be a central nervous system or the peripheral nervous system. Coming to the structure of the cell constitutes cell body, which is the main organelle, which contains all the cell organelles, cytoplasm and all those structures. And this cell body is being covered by a cell membrane, or otherwise also known as plasma membrane. And this, within the cell body, there is presence of cytoplasm. Cytoplasm is a fluid gel-like substance in which the cell organs are being present. And then comes the cell organelles, as we have already discussed. It is the ribosomes. It could be a membrane-bound organelles or a membrane-limiting organelles. And then comes the nucleus, which is a central part of the cell and which looks after all the functioning system of the entire other organelles. Starting with the plasma membrane, it is actually a main function of it is to protect. It acts as a protective sheath where it covers the cell from covers the cell's internal parts to not have any contact with the external environment, as well as the external environmental substance or the molecules. It prevents them to enter into the cell. So it has a protective function. Then comes the semi-permeability. It means semi-permeability is selective, making the membrane selectively permeable. That is, it allows only certain molecules to enter into it, which are only permeable to the membrane. Then it is usually made up of 3 layers, so it is known as trilaminar layer, the outer layer, inner layer, and the interior layer. And main structures which are present in the cell membrane are, it constitutes of lipids, proteins, and carbohydrates. Coming to the lipids of the cell membrane, usually they are these lipids, they are arranged in 2 layers. That is, they are arranged in such a form that the head of the phospholipid molecule, it is hydrophilic. That is, it is present in the outer as well as in the inner layers. This hydrophilic means it allows only water-soluble elements or molecules to enter into it. Then comes the interior part, that is the tails, which are arranged together. That makes up the hydrophobic, which is present in the middle layer, that is in the interior part. And this does not allow the water-soluble molecules into it. This is a picture showing the representation of the extracellular and the intracellular part, which is formed by 2 layers of the phospholipids. The hydrophilic head, that is the water soluble elements which can enter into it, can be seen. And then comes the hydrophobic tail, where these water soluble, it does not allow any water soluble elements into it. Next, coming to the detailed structure of a plasma membrane, which can be seen shown made up of 2 layers of a phospholipid layer, where the heads can be seen in the external surface as well as in the internal surface, whereas then the tails of it can be seen in the middle part. These structures, usually they are embedded with cholesterol as well as certain kinds of proteins. And these proteins, they are of 3 types, that is the peripheral membrane protein and transmembrane protein as well as channel proteins. Peripheral membrane proteins, they are the proteins which are only attached to the peripheral part, that is only to the external part of the plasma membrane. So the major function of this peripheral membrane proteins is that these proteins, once they interact with any function or anything, they just dissociate from the plasma membrane and they finish their function and leave that plasma membrane structure. Then coming to the integral membrane proteins, they are so called integral membrane proteins because they are embedded within the plasma membrane throughout the thickness of the plasma membrane. So that is the reason they are called integral membrane proteins, and they can be under the integral membrane proteins there are other proteins which can be categorized. There could be cell adhesion proteins or enzymes or any other proteins which are majorly involved in the synthesis of other structures. Then coming to the channel proteins, as the name itself suggests, these are the channel proteins which are usually form channels. That is, they allow the transport of certain substances from the interior to the exterior part of the plasma membrane. So they are called as channel proteins. The other group of proteins can be seen, that is, the other groups of substances can be seen as the glycoprotein. Glycoprotein is nothing but protein which are being attached with this carbohydrate structure. And the glycolipids, they are the lipids attached with the carbohydrates. This is the structure of the plasma membrane. Coming to the functions of the plasma membrane, majorly as already explained, they are protective in function and because of the semi-permeability nature, allowing only certain substances to pass through it, which do not damage the entire structure. And then comes the absorptive. It absorbs certain molecules, which makes it to have a moist and a semi-permeable. To maintain the nature of the semi-permeability, they absorb certain molecules. Then comes the excretory. Excretory, that is undigested or unwanted waste or anything which are not required in the plasma anymore in the plasma membrane. They are being expelled out by process known as exocytosis, and this that is nothing but removal of unwanted waste by the forming vesicles which get detached from this plasma membrane. They are thrown out. That is the process of that is a function which is being done. That is the excretory function. Next comes the exchange of gases. This occurs through a process known as diffusion. It could be a passive diffusion or an active diffusion. Diffusion is nothing but transport of substances from one atmosphere to another atmosphere. And coming to the types of diffusion, that is the active diffusion, which requires the expenditure of energy. Whereas in the passive diffusion, there is no need of any requirement of any energy, or it does not have to concentrate or get stressed out. Then comes the maintenance. So plasma membrane, based on all these functions, it keeps that the cell organelle or the cell structure to helps by protecting it. So by preventing other harmful substances to enter it, or otherwise any required substances prevents its escape out. By all these functioning system, it helps in maintenance of the cell organelle and its functions as well. Coming to the cytoplasm, which is a major content of a cell, and it is usually a fluid-like substance which is present inside the cell. When this cytosol can be divided, cytoplasm can be divided into two parts. That is the cytosol and the cell organelle concentrated area. So cytosol is nothing but a clear liquid portion of the cytoplasm, whereas a cell organelle concentrated area is an area where we find the huge concentration of organelles being embedded in the area. Then it contains many organelles which are different in structure as well as different in function. And usually the cytoplasm is of two zones, that is the ectoplasm zone and the endoplasm zone. The ectoplasm zone is the zone which is present immediately, that is ecto, which means outside. It is present outside, just beneath the cell membrane. Whereas the endoplasm zone is the zone which is present within the cell organelle and which is being concentrated, so it is known as cell cortex. And there are two types of organelles which are present in the cytoplasm. They are the organelles which are bound by the limiting membrane and organelles which are not bound by the limiting membrane. That is based on the presence of plasma membrane or cell membrane. So the organelles which are bound by the limiting membrane, that is where they have a proper cell envelope or a nicely well-developed plasma membrane, cell membrane. These organelles are the endoplasmic reticulum, Golgi apparatus, ribosomes, lysosomes, peroxisomes, and mitochondria. The organelles which are not bound by the limiting membrane, they are the chromosomes, ribosomes, microtubules, and microfilaments. Microtubules and microfilaments are the structures which make up the cytoskeleton of the cell organ. So there's a picture representation showing this all the kinds of cell organelles. The center of the cell, which makes up the nucleus. it is being shown with the presence of nucleolus as well as chromatin material, which is a genetic material present within the nucleus. And then immediately attached to the nucleus is the endoplasmic reticulum, that is the rough endoplasmic reticulum, showing the ribosomes being studied on the endoplasmic reticulum. And just adjacent to it is a smooth endoplasmic reticulum, which has a smooth surface without any presence of ribosomes. And then is the substance, then is the organelle showing Golgi apparatus, which shows a sac-like substances which are being elongated tubular network being shown, and it's showing with the presence of Golgi vesicles, that is, which contains the secretory or the synthesized part which is present in the Golgi apparatus is being moved away from the Golgi apparatus to its destined area. And then comes the vacuoles, then the peroxisomes and the secretory vesicles, then the mitochondria, the most important structure or the powerhouse of the cell can be termed, and then the intermediate filaments as well as the other microtubules and microfilaments can be seen. And starting with the cell organelles, that is the endoplasmic reticulum. It is a system of interconnected network of tubules and the microsomal vesicles. So usually we find the ribosome, the endoplasmic reticulum, that we found it in a network of substances, tubular substances which are interconnected with each other, and thus they form an interconnected network of tubules. And it is a membrane bound organelle. It is being covered by proper limiting membrane or the plasma membrane. And since as we have seen the position of it, it is being majorly situated just adjacent to the nucleus. So it forms a connecting structure. It acts as a connecting structure from the nucleus to the outer cell membrane, and it connects the cell membrane as well as with the nuclear membrane. The types of the endoplasmic reticulum can be classified into rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum, as the picture shows, the presence of ribosomes within the endoplasmic reticulum. Smooth endoplasmic reticulum lacks such structures known as ribosomes. Coming to the rough endoplasmic reticulum, it is also known as ergotoplasm or granular endoplasmic reticulum because of the studded nature of the ribosomes, which gives a rough appearance. And then it shows the presence of ribosomes, and also the major function of the rough endoplasmic reticulum is in protein synthesis. As ribosomes are known as the protein manufacturing structures or organelles, these protein manufacturing organelles, they get involved, they make the involvement of even the rough endoplasmic reticulum. So finally, ultimately, these are also involved in the protein synthesis. Coming to the smooth endoplasmic reticulum, since it is smooth without any granular structures on it, it is known as agranular endoplasmic reticulum, and there are no ribosomes, so it is indirectly involved in the synthesis of the proteins rather than directly being involved. And it's also, other than proteins, it is also involved in the synthesis of the hormones, steroid hormones, as well as in the cholesterol. Coming to the next cellular organelles, ribosomes, which are known as protein factories, and they appear as granular structures because of the nature of granularity. And the composition of the ribosomes, it constitutes of 65% of RNA and 35% of protein structures. They are either seen attached. They are either in the two forms. They are available in two forms. One is either being attached to the endoplasmic reticulum, otherwise they are freely present in the cytosol or the cytoplasm. Coming to the functions of the ribosomes, they are usually seen attached to the endoplasmic reticulum, involved in the synthesis of the proteins of the hormones as well as proteins of the lysosomes and proteins of the cell membrane. And then these are the type of ribosomes which are being attached to the endoplasmic reticulum. The functions of the ribosome being attached to the endoplasmic reticulum. Whereas the free ribosomes, that is the ribosomes which are being distended within the cytoplasm, they help in the synthesis of the protein part, that is the protein part of the hemoglobin, that is the globin part of the heme, and then the protein part of the peroxisome as well as the proteins of the mitochondria. Coming to the other cell organelles, that is the Golgi body. It is also known as Golgi apparatus or Golgi complex. It is being so named because it was being identified by a person known as Camillo Golgi. So it is situated near the nucleus, that is the other side of the nucleus, as we have seen in the picture already. and it is a structure where it shows flattened sac-like structures which are being arranged in a proper format or a proper sequence. So it gives the appearance of a cisternae. So it is being called as a cisternae. So presence of singular form of a singular form of a sac is known as cisterna, and plural of this multiple sacs are known as cisternae. So coming to the functions of the Golgi apparatus, since it is associated with the endoplasmic reticulum, it helps in the synthesis of the proteins. That is, the processed or the synthesized proteins from the endoplasmic reticulum, they are transported to the Golgi apparatus with the help of a transport vesicle. This transport vesicle is so called because this vesicle is it aids in the function of only transporting the synthesized, packing the synthesized substance into its vesicle, and then carrying this substance to the Golgi apparatus. So it is called as transport vesicle. From this transport vesicle, it is being carried with the help of secretory vesicle. This secretory vesicle helps in the expelling of the synthesized substance. That is, it expels out the synthesized substance out of the cell organelle by process known as exocytosis. So ultimately, Golgi apparatus or the Golgi complex, it is involved in the function of protein transport as well as protein processing the reticular vesicles, that is secreting the proteins. There is a picture of the Golgi apparatus which shows the nucleus, and adjacent to it, next immediately attached to the nucleus is the rough endoplasmic reticulum, where we can see the ribosomes being started in the endoplasmic reticulum, and they are interconnected with the nucleus, that is to the nuclear membrane. And this rough endoplasmic reticulum, adjacent to it is the Golgi apparatus, showing the transport vesicle, that is the proteins which are being synthesized in the rough endoplasmic reticulum are being transported to the Golgi apparatus. From there, they pass to the cisternae, that is the medial part of the Golgi apparatus. From there, they move out from the exterior part of the Golgi apparatus, that is a trans phase, and from there they are traveled in a secretory vesicles, and they are being expelled out of the plasma membrane, that is to the destination. They move to the intestine place. Then the other structures are the lysosomes. They are called as vesicular organelles since they are being they appear in the form of vesicles, so they are known as vesicular organism. They are also a bilayered lipid membrane which contain two layers of lipid structures. That is, the lipids being arranged in two layers, as we have seen in the structure of the plasma membrane. Then come these lysosomes, as the name itself suggests, they are the as we divide the term lysosome, lysis and soma. Lysis is breakdown or damage of any structure, and soma means it's the body. So as the name itself suggests, they are the structures which are involved in the process of removal of unwanted waste, cellular debris or any harmful substances. And to maintain a proper function of the lysosomes, they contain certain hydrolytic enzymes. That is, they contain approximately up to 50 hydrolytic enzymes in each lysosomal body, and these hydrolytic enzymes are termed as lysosomes. So these enzymes are processed and packed into the lysosomal vesicles, where they are released and show their proper function. Coming to the function of the lysosome, that is a digestion of the proteins, carbohydrates, lipids and the nucleic acids. That is, the unwanted proteins or harmful carbohydrates or lipids or nucleic acids which are supposed to damage the cell or any part of the cell. These are being digested by the lysosome. That is, they by function known as phagocytosis. That is, they take up the cell inside, they take up the unwanted waste, they eat. That is known as cell eating or cell drinking. It could be also termed as otherwise. Then that is the other function is destruction of the bacteria and the foreign bodies. That is, the hydrolytic enzymes, they cause the lysis of these bacteria and foreign bodies. They break down these bacteria into smaller amounts, and then further they are being digested by these lysosomes or secretory vesicles. And the removal of the excessive secretory products which are present in the glands of certain cells. So any unwanted waste or any harmful substances which are unnecessary, which are not anymore required in the body, are being removed by the lysosomes. So lysosomes, they have a short lifespan, because since they have the unwanted waste within it, they have to be discharged out or removed off. Coming to the other structures, they are the peroxisome. They are covered by a membrane, so they are known as membrane-limited vesicles. And since they are derived from the endoplasmic reticulum, they are derivatives of the endoplasmic reticulum, and they constitute oxidative enzymes within them. These oxidative enzymes, they help in detoxification of peroxide. That is the toxification. Any toxic substances which are present within this within the cell organelle can be detoxified. That is, the toxic substances can be removed with the help of oxidative enzymes. That is a major function of peroxisomes, and is also involved in gluconeogenesis, which is a procedure where glucogen is being formed. So the major function of peroxisome is to, for example, giving an electration of the hydrogen peroxide, which is a toxic substance to our body. It is being broken down with the help of these oxidative substances, where they break down the two molecules of hydrogen peroxide, that is 2H2O2, into one molecule of hydrogen oxide, one molecule of water, and two molecules of oxygen. Coming to the major important structure cell organelle, that is the mitochondria, which is also known as powerhouse of the cell. It is a membrane bound organelle which constitutes two layers of membrane, that is two layers of cell membrane, that is, so it is called as bilayered membranous organelle. So the composition of the mitochondria constitutes of proteins, phospholipids, as well as RNA. And coming to the structure of the mitochondria, it constitutes of the outer membrane, inner membrane, cisternae, and the matrix, where we'll be discussing further. So there is a structure picture of the mitochondria, where they can be seen the outer are properly bounded by a membrane, plasma membrane or the cell membrane, which constitutes of two layers, that is the outer membrane, and then comes the inner membrane, and in between the outer membrane and the inner membrane we see a space which is known as perimitochondrial space. Okay. Then this inner membrane, it is folded into numerous foldings, which is known as cristae. These cristae, the major function of this cristae is to increase the surface area, since mitochondria is involved in the production of the ATP. This cristae provides a greater surface area, so that there is increased production of the ATP. And there are other structures which can be seen along with the cell membrane is the ribosomes, and also the presence of certain granules which contains the synthesized ATP particles in it. And there are even certain DNA, certain structures known as DNA, that is the deoxyribonucleic acid, which is they are the extra chromosomal DNA which are present in other than the nucleus part, where we can see it in the mitochondria. So major function of the mitochondria is to produce the energy. This energy which is being produced, that is the powerhouse of the cell, produced in the mitochondria, it can also be transported with the help of mitochondrial structures, and this transported energy is being utilized by the other cell organelles. And the other functions, the minor functions which are being performed by the mitochondria, it could be the heat production as well as the storage of the calcium ions within it. Coming to the center of the cell structure, that is the nucleus, which is known as the center of the cell organelle, and it is a main, could be say a brain part of the cell organelle, since it controls the functions of the other cell organelles within it. It is a membrane bound organelle that is properly covered by a cell membrane, nuclear membrane, together known as an envelope, and it is present in the center of the cell. The other components of the cell, they include its nuclear membrane, nucleoplasm, nucleus, and the DNA. So picture showing the nucleus, showing a nuclear envelope which constitutes the outer membrane and the inner membrane. These outer membrane and inner membrane usually protect the inner part of the nucleus from the external environment, as well as from protect the nucleolus and the nucleoplasm. Nucleolus is a structure which is present in the center, which constitutes the DNA. Nucleoplasm is a structure which can be say similar to that of the cytoplasm. Since it is present in the nucleus, it is known as nucleoplasm. And then comes the chromatin material, then the ribosomes, which are being studded on the nucleus, that is on the nuclear envelope, and it contains nuclear pores. These nuclear pores, they are approximately 3000 to 4000 per nucleus. They are usually involved in the transport of the diffusion of the molecules, so they are also known as channels for the nucleus. Coming to the nuclear membrane, it is a double-layered membrane as seen, contains the outer membrane and the inner membrane, and it is porous, showing presence of numerous pores within it for the transport of the molecules. The outer layer, as already discussed, it is continuous with that of the endoplasmic reticulum, and the space which is present between the layers of the nuclear membrane, it is continuous with that of the lumen of the endoplasmic reticulum. As we have seen, the endoplasmic reticulum forms numerous tubules. These tubules, when they are connected with the nuclear membrane, are known as lumen. And then these nuclear pores, as already discussed, they have 3000 to 4000 in number, which help in transport or diffusion of these molecular substances to the nucleus which are required. And there's even exchange of materials within these nuclear pores. Coming to the nucleolus, which is another structure, it is when nucleolus is singular, it is known as nucleolus. When there are more than one nucleolus, it is known as nucleoli. And usually major content of this nucleolus, it contains of RNA, that is a ribonucleic acid, a kind of nucleic acid, and proteins. It is not being covered by any limiting membrane, that is, it is not covered by cell membrane or plasma membrane, so it is not a membrane, it is a membrane limiting organelle. Then the formation of the nucleus, majorly it is involved in the nucleus itself. Nucleolus is involved begins from the nucleus itself. The cytoskeleton, which is a major backbone of the cell organelle, could be said. It is a complex network of structures, and this complex of network of structures, it helps in determining the size as well as the shape of the cells. So it constitutes of majorly three components, that is the microtubules, intermediate filaments, and the microfilaments. So microtubules are the structures. They are known as tubular organelles because of their characteristic physical appearance, and they measure approximately 25 nanometers in diameter, and they are majorly contain a tubular protein which helps in binding all these tubules into a bundles, and so they are termed as microtubules. Since they are arranged in bundles, they are known as microtubules, and the major function of it is to provide the structural strength to the cytoskeleton or the cell organelle. It also helps in transport system. These microtubules, they can be seen majorly in cell division. Either it could be a mitotic cell division or a meiotic cell division, where we see that the chromosomes along with the centromere, they are seen attached. They are held in positions with the help of microtubules. So these chromosomes, they are being dragged to the respective poles with the help of microtubules in a cell division, that is mitotic or meiotic cell division. Coming to the intermediate filaments, they are so named intermediate filaments because they are of a diameter which is in between the diameter of the microtubule and the microfilaments. They approximately measure 10 nanometers in diameter, and the major function of it to maintain the shape of the cell. And there are certain type of intermediate filaments, that is the cytokeratins. These cytokeratins, they are grouped into two groups since they are available in huge amounts, that is, there are 30 types of cytokeratins. They make up two groups of cytokeratins. Then comes the other group of intermediate filaments, that is the vimentin, neurofilaments, laminin, and nestins. Coming to the microfilaments, they are usually a long non-tubular structures. They are not present in the form of tubules, as we have seen in case of intermediate filaments and the microtubules. They are available as well as filamentous form, that is thin thread-like structures. So they have a diameter of 6 to 8 nanometers, approximately say about 7 nanometers in size. So they are the least compared to the other two cytoskeleton structures. And they majorly provide the structural strength, that is, they resist the pressure being absorbed, and as well as maintain the cellular shape of the cell and help in the movements, that is, they provide the locomotion to the cell organelles. Coming to the functions of the cytoskeleton, as already discussed, they provide the mechanical strength, structural polarity, that is, in case of cell divisions, help in the movements of the chromosomal daughter chromatids or the parent chromatids to the opposite respective poles, as well as in the motility, that is, in the movement of any secretory products or secretory vesicle to the destined position, and in maintaining the cell's shape based on the positioning of these cytoskeleton structures. The shape of the cell is being decided, and then also helps in intracellular transport of organelles, that is, within the cytoplasm, the movement of the organelle is based on these cytoskeletal structures. Coming to the next topic, that is the cellular junctions. For this is also known as membrane junction or intercellular bridges, it can be termed, and it is so called because these cell junctions, they maintain the relation between adjacent cells as well as maintain the relation between the cell as well as to the extracellular matrix. So they are classified into three types, that is the occluding junction, communicating junction, as well as the anchoring junctions. Coming to the occluding junction, they are known as the tight junctions. These tight junctions, they are known as occluding junctions because they form a tight gap. They form a tight junctions, that is they bound the adjacent cells so tightly that they do not allow the transport or exchange of any substances between the adjacent cells. So they are known as tight junctions or occluding junctions. The major function of it is by bounding tightly the adjacent cells, they usually share the strength. They share the strength of the adjacent cells as well as share the external stress being existing on the superior part of the cell. So by this they provide the strength and stability to the tissues. And this could be given an example of renal tubular epithelial cells, intestinal epithelium, as well as in the brain, which constitutes which helps in maintaining the blood-brain barrier. So picture showing the tight junction where the transmembrane proteins, that is the proteins which are present in the adjacent plasma membranes of different cells, they are bound tightly and they prevent the movement of other structures or other molecules to enter within or out of the cell. And thus an intercellular space can be seen in between these occluding or tight junctions. Coming to the gap junctions, it is also known as nexus or an intercellular connection between two cells, that is the adjacent cells when they are bound tightly but in only a certain area, that is say about in the center of a cell, when there is connection being maintained with the help of channels or anything, then it is known as gap junction. That is, they provide gaps or channels for the movement of only certain substances, and this gap or this channels is being created by other protein known as connexins. These connexins together, they are of the adjacent cells, say about 3 from one cell and 3 from the other cell, together they bound together and they form connexon. That is, in this connexon there are 3 or 6 in number. That is, if we divide them from the 2 cells, they are 6 in number, and they provide channels. That is as observed, they are the darker greener part which can be seen, that forms the channels, that it provides the transport system for the exchange of substances or molecules between the two adjacent cells. So this forms a gap junction. Then comes the adherence junction. They are also known as zonular adherence or macular adherence. Since they adhere, that is, they get attached to each other, they are known as adherence junction. They are usually attached with the help of 2 proteins, that is known as the actin proteins and the cadherin proteins. So the actin filaments, that is, which are present in the transmembrane between the adjacent, they are present on the transmembrane, that is the adjacent plasma membrane structures, or 2 different structures of the plasma membrane, they become swollen or they form patch or plaque-like structures with the help of this actin filaments. Okay, from there these patches, then the cadherin, the cadherins, that is a glycoprotein, transmembrane glycoprotein, they get extended towards the adjacent structures, towards the against each other, and they get adhered to each other, that is, they get attached to each other and form this adherence junction. So this is being seen usually in the epithelial linings in the form of continuous adhesion. Just it is being observed just below the tight junction or the occluding junction. This makes the adherence junction. Coming to the focal adhesions, that is the cell to matrix adherence, matrix junctions, that is, they form adhesions only in focal areas, that is, say about basal epithelial layer being attached to the basal lamina in only certain areas, only selective areas where nutrition is required for the epithelium area is being passed through this focal adhesion areas, and that is being provided by the connective tissue. Which contains the blood vessels, fibroblasts, and even collagen tissue. They provide the nutrition to the overlying epithelium area. So they are termed as focal adhesion. So the focal adhesions, they are usually maintained with the help of 2 proteins, that is the actin filaments as well as the integrins, which helps in maintaining the cell to connect to the extracellular matrix as well as to its adjacent structures. Coming to the desmosomal junctions, this desmosomal junctions are the junctions which are present usually adjacent to the amongst the adjacent cells. That is, adjacent cells or a group of cells which maintain contact with each other is with the help of this desmosomal junctions, which means desmo means maintaining a proper connection between adjacent cells. So it connects the intermediate filaments of the adjacent cells. So this connection is being maintained with the help of intermediate filaments which are present within the cell. So adjacent cell membranes here in desmosomal junction, similar to that which we have seen in anchoring junctions, we see that the adjacent cell membranes, they thicken. That is, they form plaque-like structures which appear as spot-like patches or also known as plaques. They provide the major area which resists the stress being absorbed, and then they release to the adjacent structures, adjacent cells with the help of other structures known as transmembrane glycoprotein known as cadherin. This cadherin, it forms a junction between the adjacent structures where these transmembrane glycoproteins, they fuse of the adjacent structures. That is, they intermingle with the adjacent cells, and they provide such an area that the stress is being equally distributed between both the cells. So the intermediate filaments are attached with thickened patches. So that is the keratins, that is the intermediate filaments, they attach with the other adjacent cells. Coming to the hemidesmosomes, it is also known as half desmosome because since as we have seen in desmosomes, there is involvement of both the cells. But here in hemidesmosome, as the name itself suggests, hemi means half. That is only one cell which is being involved. That is, only one patch will be formed, and this patch connects with the adjacent layer, that is to say about the basal lamina or the extracellular matrix, with the help of its intermediate filaments. That is, only the intermediate filaments of the cell are being extruded from outside the cell to the basal lamina. So it is known as half desmosome. It is majorly seen as a junction between the cell and the matrix, but not between the adjacent cells. And then it connects the intermediate filaments of the cells to the extracellular matrix, and there's thickening of the membrane of the only one cell. So the major protein involved in the hemidesmosomal junction is the integrins. So there's a picture showing the cellular junctions where first junction, that is a gap junction, which can be seen, which is formed with the help of connexon. And then comes the adherence junction, which is being maintained with the actin filaments and the cadherins. And then comes the tight junctions, where they are tight occluding junctions where there is no gap between them or no even any space between them even for an air to pass through it. And then comes the desmosomal junction, that is which can be seen with the plaques being formed from them, the transmembrane glycoproteins which are being helping in maintaining adherence between the adjacent structure with the help of cadherin protein and the keratin protein. So this finishes the cellular junction. Next, coming to the nucleic acid. Nucleic acids are structures or organelles which are present in the form of nucleotides. So numerous nucleotides together, that is a polymer of nucleotides, forms a nucleic acid. So one nucleotide, it is a basic biological molecule of an organism. It is usually constitutes of nitrogen base. That is, nitrogen base, it could be a purine or pyrimidine. Purine means under purine comes the adenine and guanine, under pyrimidine comes the thymine and cytosine. So this nitrogen base along with a 5-carbon sugar, that is, it could be a DNA or RNA, along with it a phosphate group, constitute a nucleotide. So a single nucleotide consists of nitrogen base, 5-carbon sugar and a phosphate group. So nucleic acids could be of are of two types, that is a deoxyribonucleic acid or the ribonucleic acid, that is a DNA or RNA, which are being denoted as. So one DNA, that is, it constitutes of two chains of nucleic acid. So it is known as double-stranded element. Whereas RNA, it is a single-stranded chain, that is, it constitutes of only one nucleotide chain, that is one polynucleotide chain. So this is a picture showing a DNA and RNA. RNA showing only a presence of single strand, that is the presence of only the one strand of nucleotide chain, which constitutes of cytosine, guanine, adenine and uracil. Adenine and guanine constitutes a pyrimidine, and whereas cytosine and uracil form the pyrimidines. Coming to the DNA, that is, it constitutes of two structures of the DNA polynucleotide strand, which are interlinked with each other and forming a helical-like structure, that is helices, which twine around each other. So they constitute pyrimidines and purines. The pyrimidines constitute the cytosine and thymine, and the purines constitute the adenine and guanine. Then the DNA, the double helical structure is being bound to each other with the help of hydrogen bonding. So let's discuss now the DNA structure. Usually DNA is a nucleic acid. So it is present in the chromatin. Chromatin, that is which forms the chromatin together, chromatin threads or chromatin filamentous structures, they together form a chromosome. This chromosome is present in the nucleus, and this nucleus is ultimately situated in the center of the cell. So DNA, we find this nucleic DNA, which is a nucleic acid. It is present in the chromosome. It is which handles or which usually holds the genetic material of the body. So this is about the DNA. Then next, coming to the RNA. Usually RNA, which is a ribonucleic acid, kind of nucleic acid, which is usually seen in the nucleolus as well as in the ribosomes, it can be seen. So this RNA is majorly of three types, but the other RNA which has been found is a mitochondrial RNA. It's so called mitochondrial RNA because of its presence in the mitochondria. It's called as mitochondrial RNA, and along with it, it functions in a similar way. Then coming to the messenger RNA, the RNA which could be say a messenger RNA. It is so called because it carries the messages. It could be called as a postman, since it carries a message which has to be conveyed for the genetic information or as well as for the any transformation of the energy. It is called messenger RNA. In the messenger RNA, usually this mRNA, being denoted as mRNA, it carries the information which is required for the hereditary characters to be transformed, or the genetic information which is supposed to be transformed from one area, from one region to another region. Usually mRNA, the major function of the mRNA is to assemble all the ribosomes for protein synthesis. So it assembles the ribosomes which are required based on the genetic information. This genetic information could be in the form of codons, that is the codons that is in a sequence of nucleic acids or sequence of base pairing. This message is being carried, and according to based on this mRNA, the ribosomes they get arranged, they get assembled together at one area. Once these ribosomes are being assembled at one area, this rRNA, as the name itself suggests, ribosomes, that is a ribosomal RNA involved in the protein synthesis, it starts synthesizing the proteins, where it assembles the amino acids, that is the building blocks of the proteins. It assembles the amino acids together, and they together constitute a protein. That it creates a protein. Then the transfer RNA, this transfer RNA, it transfers these proteins or the ribosomes from one area to another area, which are required for the further synthesis of the proteins and further continuation of the genetic information which has to be transferred from one area to other. This completes the RNA. So this finishes the topic of cell. Thank you.